Now we've gone through, we've figured out the parts of a chromosome. We've figured out the sister chromatids versus the centromere versus the kinetochore. Now we're going to take the individual chromosomes and put them in context of the whole karyotype, the whole genome, the whole bit of chromosomes. The whole point of this lecture is to give you an idea of what a picture of all of the chromosomes look like and what information it can tell us, how we can identify a normal human karyotype. Every species has a specific number of chromosomes associated with it. Humans have 23 homologous pairs, which means how many chromosomes do we have? 46. Fruit flies have 4 homologous pairs, which means how many chromosomes do they have? 8. These particular frog, it looks like the big frog just sort of dropped the little frog, doesn't it? It's pretty bad. That's actually a female frog and a male frog. It's called a sexual dimorphism. Usually females are so much bigger than males, it's not funny. Not the way it necessarily works in humans, but in most animals, the female's really big. Male, teeny tiny. This particular group of frogs has a total of 20 chromosomes because it has 10 homologous pairs. So every eukaryotic organism has a specific number of chromosomes. We humans have 23 unique chromosomes, 23 homologous pairs that lead to 46 chromosomes. Most eukaryotic species will have 2 chromosomes that determine gender. For us, it's the X and the Y. For butterflies, it's the W and the Z. And then, of course, we get into the weird ones like bees and crocodiles and clownfish. But for now, let's just talk about X and Y. For us, if you have 2 X chromosomes, you're female. If you have an X and a Y, you're male. That is not to say that the instructions for how to build a penis are on the Y chromosome. They're actually somewhere else in the genome. They're somewhere else in some other chromosome, but they're not necessarily right there. This is more regulatory. Similarly, secondary sexual characteristics of females are not on the X chromosome. How to build a breast is not there. It's somewhere else. Aside from the sex chromosomes, all the rest of them are called autosomes. So anything that's not a sex chromosome is an autosome. So we have sex chromosomes, we got autosomes. The autosomes make up the vast majority of the instruction manual to make you. So if you look at this image, all of these chromosomes have on them, are made up of DNA. Really, really tightly coiled DNA. And all of that DNA on each chromosome will have certain genes on it, polypeptide encoding regions. So if we were to be able to read this, you would have the instruction manual for how to build every single protein in your body. And these are in every cell of your body. So you have a set of instructions in every single individual cell in your body that tells you how to build you. Now, that's just kind of cool. Not only do you have one set of instructions in each cell, you actually have 2. You've got 2 full sets of chromosomes in each cell except for your sperm and your egg. Oh, I'm sorry, your sperm or your egg. Most of it, I don't want to know if you're producing both sperm and eggs. That's a you thing. Most people are producing sperm or eggs. When you're producing 2 sets, 2 complete sets, you are called a diploid or a diploid. Di meaning 2. So here at the top, you've got one unique chromosome. Red, blue, yellow. Down here, though, we're showing 2 red, 2 blue, 2 yellow. These are diploids compared to that one, which is haploid. N versus 2N. That N is just a variable. It's just like algebra. So we call it, say, 2N and N, where N equals the number of unique chromosomes. N equals the number of homologous pairs. So if I was to say there are 4 unique chromosomes in flies, how many total chromosomes are there? If there are n unique chromosomes, what's 2n? 8. Yeah, we did this problem already, in case you missed the math, or you needed your calculator. Humans, we've got 23 as our haploid number, 46 as our diploid number. By the vast, vast majority of the cells in your body, I cannot stress this enough, are diploid. You have 2 complete sets of chromosomes. What are the 2 exceptions? Eggs and sperm. Sex cells. Sex cells are weird. We're going to devote another lecture farther down just to them. Sex cells are going to be unique. They're going to be haploid. They're going to have n number of chromosomes. Now, that's going to be important for later, because you've got an egg and a sperm, right? They're haploid. N chromosomes and N chromosomes. I don't want to spoil anything for you guys, but I hope you understand how fertilization happens. If not, we can talk in my office. Because I've had to do that with students before. So you have a sperm, you have an egg. They come together and fuse to form what's called a zygote. N plus N equals what? 2N. So you go from haploid to diploid. That's why we do this whole sex production thing, sex cell production thing. In eukaryotes, if we're passing cells on using mitotic cell division, if we're making exact copies, they're going to be inherited in full sets. Scientists who study this are known as cytogeneticists. They're the ones who look at chromosomes during cell division. And they get some cool images. The images they get are called karyotypes. Now, this is not a picture of the nucleus. This is not what chromosomes do in the nucleus. Literally, when they started doing this, you take a picture of the cell while it's dividing, and you'll be able to see the chromosomes, the individual chromosomes. In the 1960s, when they did this, they used film. I know, crazy, right? Do you guys remember film? You'd have to go and get it developed at Walmart or something. You're like, I wonder what's on there. I found a camera, like a disposable camera from when I was, 17 or 18, and I was like, one, it took me a while to find a place I could get it developed. But I got it developed, and I was like, one, there were a whole bunch of pictures of thumbs or dark things, because it was like you're just accidentally taking pictures. And it was just so weird finding things. I never realized I took these pictures. Anyways, they took a picture in the 1960s of the, through a microscope of the cell dividing. Then they took a pair of scissors after it was developed and cut out all of the individual chromosomes and by hand lined them up next to each other to figure out which ones were about the same. They could see it through their banding stain patterns. They could see it by size. After they laid them out, you could start to see patterns happening. And that let these geneticists sort of figure out what might be wrong with an individual or even what gender the individual was. So this karyotype can be used as a tool. So I want you guys to be able to read karyotypes. For instance, what's the gender of this offspring? Male. How can you tell? It's got an X and a Y. Just that easy. Any questions on that? Okay, it does get harder. It gets a lot harder. Like this one. What's the gender? How can you tell? Yeah, but what else is there? Yeah, XXY. This is called Klinefelter Syndrome. This is a problem with what's known as the non-disjunction. When the sex cells are dividing, they don't divide evenly, sometimes, by mistake, which can lead to an organism that has extra chromosomes. That's like having an extra page of instructions inserted into a manual that you can't skip. You have to follow the instructions in order. Does that make sense? So this person is producing extra proteins that may change the way their body develops. With Klinefelter syndrome, you end up with physically, the individual looks female, but they're lacking secondary sexual development. So immature breasts, likely problems with menstrual cycle, short stature, mental retardation. All because of an extra set of instructions. Does that make sense?
So
Yeah.
that person will be female?
This person is female, physically. The problem is not developing in the right way. So that's the way. This happens a lot, mainly when, as women get older, their eggs don't, how can I put this? Not to blame women or anything. But what happens is women get older, the microtubules that would attach to kinetochores don't attach properly. So instead of being pulled apart, the chromosomes just pulled in one direction, so they double up in a sex cell. That leads to major problems. That's why women generally don't give birth after 40. We've gotten better at it, but the older you get before you give birth, the more likely it is that there are going to be issues with the offspring. Not unfortunately, surprisingly enough, that only is an issue with females. Typically, male sperm, they don't have that issue. Okay, here's another karyotype. Numbers are underneath. Here's the X and Y chromosome. Is it a boy or a girl? What? It's a boy. You can tell because it has an X and a Y chromosome. What's the issue with it? Take a look at this. Do you see anything wrong? Chromosomes aren't where, well, take a look at chromosome 9. What's happened here? They're not the same size. In fact, there's a whole piece missing. That's called a deletion. You're missing a piece, which means what's gone? The directions, instructions. You're not able to build enough proteins here, which means this person would not have some of the essential proteins they need to build themselves properly. 1 in 3. This is interesting. This is called a translocation. This actually won't have much of a problem for this individual because the entire set of instructions is still there. The issue is going to come when they have kids in the next generation. Pardon?
Are they just pairs?
Yeah, well, you see, they swapped pieces. So that's not going to be much of an issue there. It's going to be an issue in the next generation, and it will eventually lead to either deletion like this or duplication where you have twice as much protein. All right, take a look at this one. Not trying to ask you to sex them because they have XY, XX, but tell me about this karyotype. Do you see anything wrong with it? Number 21. What's wrong with 21? There's an extra chromosome. This is called trisomy 21, which is also known as Down syndrome. People with Down syndrome have an extra chromosome that's producing more protein product. They have an extra bit of instruction. Typically, for humans, we can tolerate extra chromosomes or too few chromosomes down this bottom row, 19, 20, 21, 22, heck, even X and Y. Anything higher up, they're too big. There's too many instructions that would be missing or too many instructions that would be picked up if they were doubled or left out. Do you guys have any questions so far?